Hi guys, chapter 22 today. We are going over the last chapter of the semester. Um, and we're going to go over this unique organ system, the reproductive system, where it is truly differentiated into two different genders, so male and female reproductive system. It is, by the way, not the first organ system having such a differentiation. Because when you think about the urinary system, at least as far as the urethra is concerned, so there's a difference there between the male and female urethra. In the case of the female urethra, it's not shared. This is exclusively used by the urinary system. But in the male urethra, that structure is being shared by two organ systems. One by the urinary system to dispose urine, to excrete urine, and the other one is used by the reproductive system to basically ejaculate sperm out of the male's body. So that male urethra is being shared by two organ systems. And in the reproductive system, it is definitely two different organ systems, male and female. Remarkably, however, some of the organs that you learn from anatomy is basically coming from the same tissues. So similar tissues, same tissues used to produce male and corresponding female reproductive organs or structures. So today we're going to focus on the functionality of that organ system. So we're going to go over how, for example, sperm are being produced. So going over the male reproductive function, so spermatogenesis, this is how the body produces sperm. And going over the female reproductive system, which is much more intrinsic, much more complicated than the male, meaning we have to go over how an oocyte is being produced. So under oogenesis, and also dealing with not only the development of the oocyte, but also highlighting the difference between sperm production and oocyte production. Sperm production, once the male gets to puberty, he starts to produce sperm for the rest of his life until he takes his last breath. So this is a daily production process. In the case of the female, she's not producing any more oocyte when she's born. So the production of these female sex cells occurs during fetal development. And once the female baby is born, she will have all the oocyte that she needs for the rest of her life without producing new ones, because those were done during gestation when she was still in her mom's uterus. So we're going to come back and talk about that main difference between the two. But in the case of the female reproductive system, we also have to go over cycles, because in the male, they don't have a cycle. The concentration of testosterone, the main male sex hormones, is relatively constant throughout the day. Whereas in the female sex hormones go through a cycle. And we have two such cycles. The ovarian cycle, so the ovaries go through a cycle, but also the uterus also goes through a cycle. So we're going to go over ovarian cycle versus uterine cycle. It's very interesting, much more complicated in the case of the female reproductive system than the male reproductive system. So with that said, let's get the ball rolling, starting with the first slide. So sperm production, the term here is spermatogenesis. Let me start the annotation process. So, oops, hello. Okay. Spermato stands for sperm, and the word genesis means to generate or to produce. So the process, the term spermatogenesis means this is a mechanism whereby the body produces, generates sperm. And when we say sperm, whether it's one sperm or a million sperm, is still sperm. There's no S at the end. This is basically, it won't change. So we go over sperm production. Let me erase this. And it all begins with a stem cell. And that stem cell, it's called a spermatogonia. When we say stem cell, as with all stem cells, this stem cell has the potential to become any type of cell in the body. So there are more than 200 different types of cells in the human body. So a stem cell can become any cell it wants, depending on the surrounding where the stem cell is located. In the case of, since these stem cells are located in the testes, they will become sperm. So if we begin with spermatogonia, then we have to start with mitosis. Mitosis, it's a cell division that you start with a diploid parental cell, 2N. 2N means diploid, D means, di means two. So this is a cell that has two sets of chromosomes. One set came from the paternal parent, one set came from the maternal parent, right? So a total 46 chromosome, 23, that cell inherit from the paternal parent, and the other 23 came from the maternal parent. So when we say mitosis, the parental cell is a diploid, it will generate also two diploid daughter cells, each of which also contains 46 chromosome, right? 46 DNA molecule. That's mitosis. So when we say mitosis also, mitosis generates identical daughter cells. So the two daughter cells are genetically identical to the parental cell, including the stem cells in the testes. So each spermatogonium, so gonium, it ends with, instead of A, it's replaced by UM. So one spermatogonium. So each spermatogonium undergoes mitosis, which is that one type of cell division that will generate two identical daughter cells to the parental cell. So both, if the parental cell is a diploid, the two daughter cell will be also a diploid. Now, that spermatogonia undergoing mitosis will generate two daughter cells. Daughter cell A and daughter cell B. They are identical, right? They're both diploid. They have 46 chromosomes like the parental spermatogonium. The difference here is the type A daughter cell remains as a stem cell. So it will not undergo differentiation, remains a stem cell. The other daughter cell differentiates. So starts the differentiation, starts the specialization. So it will become the primary spermatocyte. Again, this, remember, this is a diploid cell, okay? 2N, containing 46 chromosomes, 23 from the father, 23 from the mother. And this primary spermatocyte starts a differentiation, and it will become secondary spermatocyte. Okay? And this is done through, I'm going to write meiosis here, or meiosis M1. So meiosis 1. And when we talk about meiosis, it's a different types of cell division than mitosis, because meiosis is a reductive cell division. When you go through meiosis, you start with a diploid parental cell. You're going to generate two haploid daughter cell. And this is through meiosis 1, M1. Okay. So you have the primary spermatocyte undergoing meiosis or meiosis 1, and you will get two secondary spermatocyte. And these secondary spermatocytes are haploid. Haploid means only 23 chromosomes. Half a set of the full complement of chromosomes in a diploid cell. And then once that happens, so I'm going to erase this for clarity. Going back to blue. Each of these secondary spermatocytes on their own will go to two additional cell division. And this is M meiosis 2. Okay, 2 as a Roman numeral. Meiosis 2. So again, meiosis, so you get haploid daughter cell, haploid daughter cell, haploid daughter cell, haploid daughter cell. And these haploid daughter cells have a name which are different. This one, these two are called spermatids. So what is important is the primary spermatocyte. One primary spermatocyte produces 4 spermatids. Through 2 successive meiosis, right? So one parental cell, being a diploid, produces 4 haploid cells. And what is amazing about these spermatids, each spermatid is genetically unique. These 4 spermatids are not genetically identical. They are dissimilar from one another. Not only they're different from one another, they are also different from the parental primary spermatocytes. This is why meiosis is an important cell division mechanism to generate genetic diversity in a population. So the human species, using sexual reproduction, and through sexual reproduction relies on meiosis, and meiosis is a cellular division, a mechanism of cellular division that can generate diversity, that can generate variability. So you can introduce genetic diversity into a population of species, in this case, the human species. The reason is each haploid daughter cells resulting from meiosis 1 and meiosis 2 is genetically unique. That uniqueness, this is why you are so different from your sibling and from your parent, yet you are related to one another, right? The only way for a human being to have a full complement of genetically identical complement, like that's only possible if you have an identical twin, right? So in other words, one initial zygote splits into 2, and each one goes on to basically becoming a full, complete the development and becoming 2 human beings, yet these 2 human beings are genetically identical. We can also have another set of twins. They are called fraternal, meaning they were fertilized by, so 2 different sperms fertilizing 2 different secondary oocytes. So those are 2 different human beings. They have 2 different sets of genetic makeup that are unique to each of them. So it's like you and your sibling, right? Even though you are twins, if you have a fraternal twin, there's no difference in terms of you and another sibling, because in terms of genetic makeup, each one of you are unique. So the only way to have sharing the same genetic full complement of 46 chromosome, you have to have an identical twin. Okay? So that meiosis, being a cell division mechanism capable of generating genetic diversity, is unique for sexual reproduction. And any types of living organism, any type of species undergoing sexual reproduction will have genetic diversity in a population within that species. So any organism that do not use sexual reproduction, do not use meiosis, we have a problem. They cannot generate a lot of genetic diversity within a population. The only source to generate genetic diversity, for example, among bacteria, because they don't use sexual reproduction, is through random mutation, right? So random mutation is not as effective introducing genetic diversity in the population compared to sexual reproduction. Okay. That's a little bit out of the scope of the physiology, but I thought it was, refreshing to refresh your memory about it, because I don't know about you, but I teach anatomy. We talk about this during anatomy classes. So some of it is basically a review. That's why I'm concerned when we go over this, because all these names, primary spermatocyte, secondary spermatocyte, spermatids, were covered in anatomy already. Now, once you get to the spermatids, we're not done. The spermatids will go on to complete the process by another step. It's called spermiogenesis. So there's two words here, spermiogenesis and spermatogenesis, right? During this step here, the spermiogenesis, toward the end of spermatogenesis, spermatids become the sperm. So, and ironically, the sperm, when they are produced, they are still non-functional, right? So the site of their production, the birthplace of the sperm, is happening in a structure, a tube-like structure called seminiferous tubule. And in each testes, you have, smaller compartments, up to 300 lobules within each testis. And within each lobule, it contains one seminiferous tubule. So if each testis consists of 300 lobules, then each testis contains 300 seminiferous tubule. And each seminiferous tubule is the birthplace, the production site to generate, to produce, to synthesize sperm. And the sperm, once they're being produced, they're not functional. It takes some time for them to go through the maturation process as they transit through the duct system, through the male reproductive tract. Okay. So this is an example here. This is one cross section of a seminiferous tubule. It's coming out of the slide at you, and you cut it across and you see the inside, and the sperm are basically released into the luminal space here. Inside that red circle, it's an empty space that will receive the sperm, and that luminal side runs the entire long axis of that seminiferous tubule. And you have it here. This is the same red circle that I drew over here. Right, the sperm are being released into that luminal side within each seminiferous tubule. And we're going to come back to these cells, the Leydig cells and the Sertoli cells. They're the important critical role involving in inducing spermatogenesis. And this picture is an overview of what I just described earlier. So this is the luminal side here. Sperm will be released into here. You have the Sertoli cells. And within this area, the Sertoli cell are basically between the Sertoli cells, rather. They basically, there's a process that basically your stem cells, male sex stem cells, become a sperm. So we have here mitosis. So type A, type B, Type A remains a stem cell, and type B goes on the differentiation process to becoming a sperm, right? So here we have mitosis, but here we have meiosis. So meiosis 1 and meiosis 2. What is important is you start out with one diploid. Okay, so this is a diploid cell here, 2N, the primary spermatocyte undergoing the first meiosis, producing two secondary spermatocytes. Each one is a haploid. So this and this here is two haploid cells. And each secondary spermatocyte undergoes meiosis 2 or meiosis 2 to produce 4 haploid spermatids. And the spermatids, through spermiogenesis, become 4 sperm. Each of which also obviously are haploid cells. Keep the three-dimensional relationship between the Leydig cells and the Sertoli cells in the back burner, because we will talk about them, their importance in releasing chemical messengers to kickstart the process, inducing the process of sperm production. And they are themselves under the influence of hormones coming from the anterior pituitary gland. And the anterior pituitary gland receive another regulatory factor coming from the hypothalamus. So all of this comes back. We learned this from the endocrine system. So in this case, we're going to focus on the reproductive system of these endocrine functions. So involving, for example, GnRH, involving luteinizing hormone, involving FSH hormone, and inducing these cells to produce, for example, inhibin, inducing androgen binding protein, inducing testosterone, which is a male hormone. We're going to talk about it for the next few slides. But in the process, keep in mind the three-dimensional, the relationship between the Leydig cells here and the Sertoli cells within the testes. There's the relationship between the brain and the testes. It's called the brain testicular axis. Oops. So hormone control over testicular functions. So starting with the hypothalamus, the master switch of the endocrine system, which is a neural structure. So the hypothalamus belongs to the nervous system, and the nervous system controls the entire endocrine system through the hypothalamus. So the hypothalamus releases a regulatory factor, right? RF, which is a hormone. And this regulatory factor specifically is the gonadotropin releasing hormone, GnRH, released into the hypophyseal portal system. And this GnRH targets the APG, the anterior pituitary gland. Okay, so that's the target. So anterior pituitary gland here, GnRH induces anterior pituitary gland to release two anterior pituitary hormone. One, FSH, two, LH, follicle stimulating hormone and luteinizing hormone. Now, FSH will use the bloodstream. Remember, this is in the pituitary gland. So FSH here will use the bloodstream. So this is a bona fide hormone, and it goes all the way into the testes and target the Sertoli cells. So FSH targets the Sertoli cells, whereas LH target the Leydig cells. And in turn, the Sertoli cells, also known as sustentacular cells, secrete two additional chemical messengers, ABP and inhibin. Whereas the Leydig cells, also called interstitial cells because they are located in the interstitial space, secrete the most important male sex hormone, testosterone. This testosterone, it's a nightmare in terms of social behavior. For example, it drives, this is a main chemical messenger that increases sex drives in males. It also increases violence in males. This is the reason why males have also more aggressiveness compared to females. It also drives the secondary, sexual organs like the male appearance, deepening the voice. When a boy hits puberty, he changes his voice. That's due to testosterone. So one single chemical structure that can have drastic outcomes in terms of phenotype, right? Both external physical appearance, but also behavior-wise as well. And this is remarkable because even if the person is biologically born as a female, I'm talking about transgender here, and the female decides to become a man, she will take testosterone for the rest of that person's life in transitioning to become a male, a man, and that genetically female, biologically female, will have a sex drive as strong as a biological male, just simply because that person, the transgender, female to male transgender transition, is taking testosterone. So that's a very, very nasty, steroid hormone. This is fat soluble. So this testosterone has no problem going, across all membranes. Now, this testosterone drives sperm production, right? So in the testis, which I will talk about the next, ABP. ABP stands for androgen binding protein. Again, androgen, ANDROGEN. Androgen means male sex hormone. Binding, protein. So this chemical messenger binds to testosterone. In other words, it concentrates testosterone around the Sertoli cells, right? Remember, the Leydig cells produces and secretes testosterone. So in this case here, testosterone has two functions. It acts as a paracrine signal when it is acting on the Sertoli cells, paracrine. So in other words, it functions as a short distance chemical messenger. It is also released in the bloodstream, so it is also acting as a hormone. In this case. So testosterone, in this case, functions both as a paracrine signal and as a long-distance chemical messenger that we call hormone, because it will be released into the bloodstream, right? And we'll talk about this evidence, testosterone being released into the bloodstream, on the next slide. Now here, so let me see if I have it in your book so you can follow. So I'm referring to figure 22.8 on page 641 of your Stanfield textbook. And if you follow that picture, you're seeing once the Leydig cells are producing and releasing testosterone. So testosterone here, if the testosterone can basically target the Sertoli cells, that's the paracrine signal. But these three functions here, testosterone is acting as a hormone. The reason is because testosterone needs to be released into the bloodstream so it can have access. So it will move from the testes all the way up to the hypothalamus to inhibit the release of GnRH. This is targeting in the hypothalamus, whereas the next two, it targets the anterior pituitary, right? So testosterone affects the anterior pituitary gland. So it induces the inhibition, it prevents the anterior pituitary gland from releasing follicle stimulating hormone and luteinizing hormone. Whereas it also inhibits the hypothalamus to release the GnRH. So I'm going to just simply write hypo here for hypothalamus, okay? And you can see that on figure 22.8. So testosterone, through negative feedback, it will go back from the testes, it will go into the anterior pituitary, and it will shut down both FSH and LH release by the anterior pituitary gland. It can also go to the hypothalamus and it causes the hypothalamus, it shuts the hypothalamus, so the hypothalamus will shut down its own GnRH release every time that testosterone shows up at its door. So this is how testosterone, through negative feedback mechanism, influencing both the hypothalamus and the anterior pituitary gland from decreasing, from stopping the respective hormone release. GnRH in the case of hypothalamus, FSH and LH in the case of the anterior pituitary gland. That's for the testosterone, the negative feedback involving testosterone as a chemical messenger, both as long distance chemical messenger and as short distance chemical messenger. The Sertoli cells themselves also secrete chemical messenger. And the Sertoli cells secrete a hormone called inhibin. INHIBIN. Inhibin targets the anterior pituitary gland. The difference here is it can only inhibit the release of FSH by the anterior pituitary gland. Right? So it does not influence the release of luteinizing hormone by the anterior pituitary gland. And check this out. This is a complete, I don't know why I put it there, but that's wrong. Inhibin does not go to the hypothalamus, does not influence hypothalamic release of GnRH. Okay, so that's, and then on the next slide, it shows you that picture that I just referred to you, because you can use the book when I'm talking about this slide. So this is the picture, the figure that I mentioned earlier. So this is the picture of figure 22.8 that I mentioned earlier. So you see here the Leydig cell produces, secretes testosterone, and testosterone, this arrow, that's a paracrine signal, short distance messenger. Okay, P stands for paracrine. Whereas this arrow, testosterone is acting as a hormone. H stands for hormone. So testosterone as a hormone released into the bloodstream. So plasma here means it's being released into the bloodstream, the blood plasma. And from there, it will go up and affect anterior pituitary and hypothalamus. Whereas the paracrine signal is locally affecting the Sertoli cells, right? So paracrine affecting Sertoli cells, hormones affecting anterior pituitary and hypothalamus, as far as the testosterone is concerned. So produced by Leydig cells and released by Leydig cells, testosterone. And Sertoli cells secretes inhibin, but I also mentioned Sertoli cells secretes A, B, P, androgen binding proteins. So the presence of ABP, androgen binding proteins, it binds testosterone, it concentrates testosterone in the Sertoli cells, and the Sertoli cells induces spermatogenesis. Okay. So both FSH and testosterone induce spermatogenesis. So the more testosterone arrive, the process of spermatogenesis would be facilitated. And remember, the male produces its sperm every day, and the concentration of testosterone is basically, although it fluctuates daily, every 2 hours, but it remains stable throughout the day. And surprisingly, when the male is ejaculating his sperm, those sperms were produced um, not a few minutes ago, not even the morning, or not even the day before, not even the week before. They were produced 3 months prior, right? So the sperm that was just ejaculated were produced, were synthesized, were formed 90 days ago. And they were stored in the vas deferens, right? So the vas deferens is storage site for sperm for up to 3 months, several months. And so that's the amazing part. So by the time the guy is basically ejaculating by the third time or the fourth time, that semen is most likely very few sperm are remaining. The reason why I'm saying this is because the body cannot unload enough sperm with successive ejaculation within a few hours. So he has to wait until the next day, the very least the next day, to have a new load of sperm that can fertilize a secondary oocyte. And on another remarkable event is the volume of that sperm, of that ejaculatory volume, it's around only 5 ml, 5 cc's, like around a teaspoon's worth. And that volume, 95% of that volume, we're not talking about sperm, we're talking about secretory products coming from accessory sex glands, like the paired seminal vesicles, the paired Cowper's glands, and the single prostate. So those are 5 glands contributing to secrete 95% volume-wise of those fluids coming from these glands. And only 5% contains the actual sperm. So every 5 cc's of semen, only 5% of which contains the sperm. The rest is just seminal fluids coming from the seminal vesicles, prostatic fluids coming from the prostate, Cowper's fluid coming from the Cowper's glands, right? And within that fluid, it contains, for example, fructose, which is a monosaccharide, feeding the sperm, because the sperm needs ATP to swim. So it basically, it oxidizes, it breaks down the fructose within coming from the seminal vesicles to basically allow them to give to move, to swim from point A to point B once they've been deposited in the female reproductive tract, getting to that secondary oocyte. Another remarkable thing is when they get inside the vagina, 95% of these sperms know exactly where to go. They can sense which ovary is ovulating. So 95% of the sperm, around 300 million to 500 million sperm being deposited in the vagina, a birth canal, 95% of them will go to the correct side of the ovary that's ovulating. Even sperm, you have la la land lost sperm, the 5% would go to the wrong side of the ovary, and that ovary is not ovulating. So that's basically through chemical signaling. So they know, they sense, they know which ovary it's ovulating, that secondary oocyte, and they get there, right? Okay, so that's for my brief review about the journey of the sperm in the reproductive tract. Moving on to the next one. The process of producing oocytes. So oocytes is the female sex cell. The same process. We begin with a stem cell. In this case here, it's called oogonia. So one oogonium. The A is replaced by UM. So one oogonium, two or more oogonia. The difference here is by the time the female baby is born, she has all her female sex cells in her ovaries. So she's not producing new ones after birth. That's the main difference between male and female reproductive system when we talk about developing, producing sex cells. Again, each oogonium is a stem cell. So the stem cell divides itself by using, again, mitosis. So same process, you start with a diploid parental cell, you get two diploid daughter cells, genetically identical, right? So one oogonium remains as a stem cell, the other one becomes a primary oocyte. And the difference also is each oocyte is protected, located inside a larger structure that we call follicles. This should be a review material from anatomy. And also amazingly, once they're produced, they are not allowed to complete meiosis. So late prophase 1 here means this is part of meiosis 1. And meiosis 1 is completed only upon puberty, when the girl hits puberty and she starts the process of menstruation. So meiosis 1 is completed each month, and the primary becoming secondary oocyte. And this is an important, and this should be covered in anatomy. The secondary oocyte is the cell that gets ovulated on a monthly basis. So when you're ovulating, you're not ovulating an egg, you're ovulating a secondary oocyte. This cell has not completed its cell divisions yet, right? And it will hit meiosis 2, and then again it stops, arrested in metaphase 2. So this is part of meiosis 2. And it will complete meiosis only if the cell, this secondary oocyte, is being fertilized by a sperm. So if a sperm penetrates and ovulates a secondary oocyte, then that secondary oocyte completes meiosis 2. And once it completes meiosis 2, it has a unique name called ovum. This ovum, what is unique about this ovum is it has two sets of chromosomes. So it has the N from the female parent, and it also have another set from the male parent, right? So in other words, these 23 chromosome is within the oocyte. These 23 chromosome comes from the sperm. They are not fused yet into one nucleus. So technically speaking, the ovum contains 46 chromosomes, but there is still a, it's considered to be a haploid cell. Even though it has 46 chromosomes, it is considered to be a haploid cell. The reason is because the nucleus of the ovum, inside the ovum, you still have only 23 chromosomes. Because the 23 chromosome from the sperm they haven't been fused into one single nucleus yet, right? So that's a tricky part there, a gray zone in terms of the ovum. So if someone asks you, how would you classify an ovum? You need to explain clearly. The ovum is considered to be haploid because in its nucleus, it has only 23 chromosomes. However, the entire cell, in the cytoplasm of that ovum, you have another 23 chromosome coming from the nucleus of a sperm. So the two nuclei have not fused into one yet. Once that happens, if the two nuclei in an ovum fuse into one single nucleus, that ovum becomes what is called a zygote. Zygote. The zygote is a one cell future human being. We all start at one cell stage. That zygote is a one cell stage. It's a diploid, bona fide diploid one cell stage that will ultimately become a human being. It has all the complement 46 chromosome in one single nucleus that has the potential to become a future baby. So the next slide should have a picture going back in detail, showing you which part of cell division each oogonium goes through. Okay. The female fetus starts with up to 4 million stem cells. So anywhere from 2 to 4 million stem cells. And this is through fetal development, meaning before birth. This is BB, before birth. And this is after birth. Whereas within the afterbirth, and then over here, okay, so you see here, mitotic arrest, same thing here, it gets, the secondary oocyte stops its development. So you have here the diploid stem cells undergoing mitosis. This cell remains a stem cell. This cell kickstart the differentiation process, becoming a primary oocyte. It is still a diploid. And that primary oocyte stops its development and it remains as is. In in the fetus, and even the female baby is born, it remains as a primary oocyte until puberty. So when the girl hits puberty, this primary oocyte completes meiosis 1 to become a secondary oocyte. The other cell, it's called a first polar body, it's non-functional. It remains on the surface of the secondary oocyte. It's stuck there, but it's non-functional. All it has is just its nucleus. It lost its cytoplasm to this secondary oocyte. So this is the cell that you're ovulating on a monthly basis. And that secondary oocyte remains as is and lost through the menses if there's no fertilization. It will never complete its development. If it is fertilized, if it encounters a sperm and is fertilized by a sperm, then it will complete all its development, at which case it is called an ovum. Okay. So the ovum technically, like I mentioned earlier, is not a diploid cell. It contains both complement, the maternal chromosome and the paternal chromosome from the sperm, but they are in two separated nuclei. The maternal chromosome is in the nucleus of the ovum, and the paternal chromosome is in the nuclei of the X sperm, right? They haven't fused yet. And I mentioned earlier, once they fuse, that ovum will be called a zygote. And the zygote is the first bona fide diploid cell. So one cell stage of a future human being. So the girl, the baby, the female fetus started up to 4 million oogonia, but by the time she's born, it's down to 2 million, and by the time she hits puberty, it's only less than 500,000 oocyte, because most of them undergo what is called atresia, meaning they self-destroy themselves because lack of hormones and lack of sensitivity to hormones, especially estrogen and luteinizing hormone. And every month you lose around 1,000 oocytes a month. Only one single oocyte is selected to become the mature Graafian oocyte, follicle, sorry. So each month you lose 1,000 oocytes, so meaning you lose 1,000 follicle, and only one follicle gets selected, and that follicle is called the dominant follicle. It will become the mature follicle, which is also known as a Graafian follicle, Graafian, Graafian follicle. And that Graafian follicle kicks out the secondary oocyte. So it's ovulating that secondary oocyte. And that Graafian follicle, when it kicks out the secondary oocyte, it becomes a gland called a corpus luteum. And that corpus luteum actively secretes chemical messengers from the oocyte, not from the ovary, using the bloodstream, estrogen and progesterone, going all the way down to the uterus and prepping the uterus for implantation of a future human being, right? So the corpus luteum remains active for around 10 days. If there's no fertilization, then the corpus luteum starts to disintegrate and it becomes the corpus albicans, which is a scar on the surface of the ovary. And remarkably, interestingly, if you have a microscope and you can look at the surface of the ovary of a female patient, you can estimate her age by counting on these the present scars on the surface of each ovary. they look like a crater on the surface of the moon, right? So each scar represents one ovulation event. So assuming that the girl starts ovulating around age 12, let's say, and you count the number of scars on each ovary, you can estimate the age of the patient, right? Give or take 2 to 3 years, assuming, not all girls start puberty at 12, obviously, but it gives you an idea of how old the patient is just by looking at the surface of her ovaries. And also, the process of maturation of the follicle becoming the prominent, the dominant, the Graafian follicle is completely random. I know that there are rumors out there, anecdotal rumor that say, okay, your ovaries alternating to ovulate on a monthly basis. That's not really true. It's completely random. It's this, statistically speaking, it's completely random. And if there's a pattern, it is not known yet as of today. But if you know, share that with me. I would love to know. Okay, but right now, as of now, the maturation of the Graafian, that dominant follicle, is completely random. It's a random process. There's no pattern attached to it. And it is also, it's a rare event that yes, the woman can ovulate not one but two different secondary oocyte. Also, it's around 1 to 2% in the population. So it's a rare event. Now, talking about the menstrual phase, and when we talk about menstrual phase, there are two cycles. Sorry, menstrual cycle. So there are the ovarian cycle and the uterine cycle. Let me take this out of the way. So if you look the ovarian cycle, let me look up the page here. So I will refer you to the figure. Ovarian cycle, this description on this slide corresponds to figure 22.15, 22.15, page 650, page 650 in your Stanfield textbook. And you can clearly see the follicular phase and the luteal phase. The ovulation is smack in between the two phases. And what is remarkable is, although I say 20 here, it can be up to 25 primordial follicle. And remember, each follicle contains only one oocyte, and each month only one selected to continue to meiosis one. So this one selected follicle is down here, the mature follicle, the dominant follicle, the Graafian follicle. This follicle, this is the one that ovulates, the one that kicks out, ejects the secondary oocyte. The luteal phase is important. Once the Graafian follicle ejects a secondary oocyte, the Graafian follicle becomes a gland, corpus luteum. Okay. Again, follicle, Graafian follicle, when it ejects a secondary oocyte, kicks out a secondary oocyte, it becomes the corpus luteum. And that corpus luteum, this is important, secretes progesterone. This is a hormone. The progesterone will leave the ovary and will use the bloodstream to get all the way down to the uterine wall, the wall of the uterus, right? And that progesterone is basically prepping the uterus to receive the fertilized oocyte, the future human being. And that corpus luteum I mentioned earlier remains active for around 7 to 10 days. Now, if there's implantation, meaning that zygote, it will travel from the fallopian tube because the fertilization occurs in the fallopian tube. So the sperm meets the secondary oocyte during its transit in the fallopian tube, and that fertilized secondary oocyte, the zygote, takes around a week, right? The journey takes around a week, 6 to 7 days, from the fallopian tube all the way down to the uterus. So it implants itself. So if there's implantation, the endometrium, which is the mucous membrane of that uterus, and ultimately later by the future baby, it releases a hormone called HCG, lowercase h, uppercase C, uppercase G. And this HCG, human chorionic gonadotropin, will go back to the ovary, and it keeps the corpus luteum active. So this is a chemical system. So the human chorionic gonadotropin produced by the endometrium and later by the placenta make sure that the corpus luteum remains active, especially for the first 8 weeks, because the corpus luteum maintains the embryonic development, so maintains the embryo for the first 8 weeks. Right? So embryonic development is basically mediated by the corpus luteum. And that's the first 8 weeks. The reason is because the first 8 weeks, the placenta is non-functional. After 8 weeks, the placenta takes over. And that's where fetal development begins, right? So the placenta makes sure that the fetus completes the development and becoming a functional, living baby. But the first 8 weeks during the embryonic development, that embryo is being fed by the corpus luteum, is being maintained by the corpus luteum, whereas the placenta maintains the development of the fetus from week 9 to week 39. So this is the picture that I mentioned earlier. Now, most of the student this picture, same thing with anatomy. It's a lot of ground for confusion. Step one, stage 1 all the way to stage 9 here remains in place. It doesn't move like this. It shows you like the cell is moving through it. No, it's not. It's all starts here, right? So at this location, it starts at 1, it moves into 2, 3, 4, and 5. It stays here. 6, 7, 8, 9 stays here also. So the primordial follicle was here. This particular primordial follicle, the selected one that becomes a Graafian follicle, was this location. And one that is being selected to become the dominant follicle, the Graafian follicle, it kicks out the secondary oocyte. This is a secondary oocyte, okay? So the remaining part here becomes a gland. We call that corpus luteum, this part, for 10 days, after which, if there's no implantation, it becomes a scar on the surface of that ovary, which is here, called corpus albicans. So the book has to do this to show you the different stages that the development ultimately from primordial follicle becoming the Graafian or Graafian follicle, okay? And you lose, every month there's like 15 to 25 follicle competing, and only one is basically being selected because that dominant follicle develops faster than the other competing follicles in the process of becoming that Graafian follicle. And each month you also lose, in addition to these 25 competing follicles, you lose an additional 1,000. So you lose around 1,000 follicles per month. But you have more than enough for your reproductive fertile life before you hit menopause. So that's the story for the ovarian cycle. Moving on to the other cycle, the uterine cycle. The difference between the two, the uterine cycle is divided into 3 phases. So I'm not going to talk about this corresponding to the picture on, so figure 22.16. page 652. So it has 3 phases: menstrual phase, proliferative phase, and secretory phase. The menstrual phase lasts up to 5 days. Again, you know, I hate to generalize like this. From it, anywhere from 5 days to more than a week, depending on the flow of each female individual. But what we are talking about here is the entire superficial layer of epithelium from lining the uterus is completely destroyed, completely shed off. And that superficial epithelial layer is called a stratum functionalis. This is from anatomy. And that stratum functionalis is basically epithelial cells that get shed off, completely destroyed. And then the deeper layer called a stratum basalis rebuilds that stratum functionalis every month, right? So after the menstrual phase, we have the proliferative phase. This is where estrogen kicks in and induces the development of that follicle to produce that Graafian follicle, the mature dominant follicle that contains a secondary oocyte to be ovulated. And during this phase also, it reveals the stratum functionalis. And this is done by the stratum basalis, the deeper layer, right? So the stratum basalis in the wall of the uterus reproduces, rebuilds the stratum functionalis. And this also has progesterone receptors to basically, it responds to progesterone. So it's prepping, the progesterone is prepping the lining of the uterus to be ready for the implantation of a future human being. And the secretory phase, progesterone comes from the corpus luteum. This is once ovulation occurs, right? So for the next 10 days, 7 to 10 days, the corpus luteum secretes progesterone, and the progesterone goes all the way down to the uterine wall and prepping itself. So makes the uterine wall to be ready to receive that zygote. By this time, it's no longer a zygote. It has around 64 cells or something like that, and it will implant itself into the wall of the maternal uterus. So in other words, progesterone is basically making sure that mucous membrane lining of the uterine wall called the endometrium is ready for implantation. And this is a summary. You have your ovarian cycle lined up with the uterine cycle. So the ovarian cycle, you have only two phases, follicular phase and luteal phase. And what is remarkable, again, the follicular phase is always 14 days. Always. Very little changes between one woman to the next. And you're going to say, dude, my cycle is not 28, right? Like the book says 28. Again, it's true. It's you cannot generalize like that. So each female patient or each woman has her own unique cycle. So the reason why you can have variability is this occurs during the luteal phase. This can be different than 14 days. So if your cycle is 30 days, it means the follicular phase 14, the luteal phase is 16. If you have cycle at 26 days, then the follicular phase still 14, the luteal phase will be 12. If you have 33 days, then 14 for the first and the balance for the rest. So what allows the variability between one woman to the next in terms of her menstrual cycle is due to the variability of the luteal phase, not of the follicular phase. So the follicular phase is relatively constant from one woman to the next. So the menstrual cycle, the menstrual phase starts at day one. So the very first day that you have your menses, that's your cycle. That's when it begins. So again, this for the first 5 days, what's going to happen is all these blood vessels, without chemical signaling, they start to shrink, they start to constrict. And once they constrict, there's no more oxygen coming in, there's no more nutrients coming in. And the superficial layer here, the epithelial layer of the stratum functionalis start to die off, and they start to detach from the stratum basalis, the basal layer. Okay? So as soon as the corpus luteum is not receiving the HCCH, human chorionic gonadotropin, then it stops secreting estrogens and progesterone. And once the uterine wall is not receiving progesterone, then the blood vessel starts to constrict, nutrient arrival stops, carbon dioxide accumulates, the cells die, and once the cell die, they start to detach. The stratum functionalis starts to detach, and break open the vascularization, the blood vessels, tear off. This is where blood comes out. So this is the menstrual flow. And that menstrual flow contains blood and also a mixture of epithelial cells coming from that stratum functionalis, the superficial layer of your endometrium that's being shed monthly basis. Once at the end of the menstrual flow, the menstrual phase, the proliferative phase starts all over again to build that stratum functionalis, SF. So this here is readies the endometrium for implantation. And once the future baby, the fertilized oocyte, implants into the endometrium will send back HCG to the ovary to maintain that corpus luteum. And the corpus luteum gets functional, remains functional at least for the next 8 weeks, maintaining that developing embryo until the placenta is mature enough to take over. Okay. And here is just the relationship between the hormones in the bloodstream. And I ask you here, meaning of the LH surge. This LH surge here induces ovulation. This induces ovulation. And what causes the LH surge? You can go in your book and look it up. And if you don't know, you can always ask me on the discussion board. And what I want you guys to go back to, and there are figures that are important to help you understand all of this, is figure 2218, 2219, and 2220. That shows you the relationship between these hormones during the female reproductive cycles. I will not ask any of that because I haven't covered that, but that's for personal satisfaction to complete the picture on this. Due to time constraints, I cannot go over this, and this is not ideal to explain it by Zoom conference. I would have gone through it in a regular semester using a whiteboard, but since I'm not doing it with you, I'm going to give you guys a break. But you should, I strongly recommend that you read, you go over figure 2218, 2219, and 2220 to complete the picture, allowing you to understand it, to appreciate this on a much deeper level than what I'm covering here. But as far as assessment-wise, I will not assess you on information contained, the details from these three figures, 22.18, 2219, and 2220, except when it's relevant that I cover during lecture, okay? And then on that note, I will officially end the last recording for Bio32. I wish you well, remain healthy, stay safe, study well, and make me proud, okay? Take care.